Carnivorous plants are plants that derive some or most of their nutrients from trapping and consuming animals or protozoans, typically insects and other arthropods. Carnivorous plants have adapted to grow in places where the soil is thin or poor in nutrients, especially nitrogen, such as acidic bogs and rock outcroppings. Charles Darwin wrote Insectivorous Plants, the first well-known treatise on carnivorous plants, in 1875. True carnivory is thought to have evolved independently 6 times in 5 different orders of flowering plants, and these are now represented by more than a dozen genera. These include about 630 species that attract and trap prey, produce digestive enzymes, and absorb the resulting available nutrients. Additionally, over 300 protocarnivorous plant species in several genera show some but not all of these characteristics. Trapping mechanisms. 5 basic trapping mechanisms are found in carnivorous plants. Pitfall traps trap prey in a rolled leaf that contains a pool of digestive enzymes or bacteria. Flypaper traps uses sticky mucilage. Snap traps utilize rapid leaf movements. Bladder traps suck in prey with a bladder that generates an internal vacuum. Lobster pot traps force prey to move towards a digestive organ with inward-pointing hairs. These traps may be active or passive, depending on whether movement aids the capture of prey. For example, Traphiophyllum is a passive flypaper that secretes mucilage, but whose leaves do not grow or move in response to prey capture. Meanwhile, sundews are active flypaper traps whose leaves undergo rapid acid growth, which is an expansion of individual cells as opposed to cell division. The rapid acid growth allows the sundew tentacles to bend, aiding in the retention and digestion of prey. The sundew species Drosera glandulidra employs a unique trapping mechanism with features of both flypaper and snap traps. This has been termed a catapult flypaper trap. Pitfall traps. Pitfall traps are thought to have evolved independently on at least 4 occasions. In general, they are phytoautomata, water bodies collected or secreted into specialized containers, and ultimately held by plants for various functions such as in particular, the trapping and digestion of prey. The simplest ones are probably those of Helianthera, the marsh pitcher plant. In this genus, the traps are clearly derived evolutionarily from a simple rolled leaf whose margins have sealed together. These plants live in areas of high rainfall in South America such as Mount Roraima and consequently have a problem ensuring their pitchers do not overflow. To counteract this problem, natural selection has favored the evolution of an overflow similar to that of a bathroom sink. A small gap in the zipped-up leaf margins allows excess water to flow out of the pitcher. Helianthera is a member of the Sarraceniaceae, a New World family in the order Ericales. Helianthera is limited to South America, but the family contains 2 other genera, Sarracenia and Darlingtonia, which are endemic to the southeastern United States and California respectively. Sarracenia purpurea subsp. Purpurea can be found as far north as Canada. Sarracenia is the pitcher plant genus most commonly encountered in cultivation, because it is relatively hardy and easy to grow. In the genus Sarracenia, the problem of pitcher overflow is solved by an operculum, which is essentially a flared leaflet that covers the opening of the rolled leaf tube and protects it from rain. Possibly because of this improved waterproofing, Sarracenia species secrete enzymes such as proteases and phosphatases into the digestive fluid at the bottom of the pitcher. Helianthera relies on bacterial digestion alone. The enzymes digest the proteins and nucleic acids in the prey, releasing amino acids and phosphate ions, which the plant absorbs. Darlingtonia californica, the cobra plant, possesses an adaptation also found in Sarracenia psittacina and, to a lesser extent, in Sarracenia minor. The operculum is balloon-like and almost seals the opening to the tube. This balloon-like chamber is pitted with area low, chlorophyll-free patches through which light can penetrate. Insects, mostly ants, enter the chamber via the opening underneath the balloon. Once inside, they tire themselves trying to escape from these false exits, until they eventually fall into the tube. Prey access is increased by the fish tails, outgrowths of the operculum that give the plant its name. Some seedling Sarracenia species also have long, overhanging opercular outgrowths, Darlingtonia may therefore represent an example of neoteny. The second major group of pitcher plants are the monkey cups or tropical pitcher plants of the genus Nepenthes. In the 100 or so species of this genus, the pitcher is born at the end of a tendril, which grows as an extension to the midrib of the leaf. Most species catch insects, although the larger ones, such as Nepenthes raja, also occasionally take small mammals and reptiles. Nepenthes bicalcarata possesses two sharp thorns that project from the base of the operculum over the entrance to the pitcher. These likely serve to lure insects into a precarious position over the pitcher mouth, where they may lose their footing and fall into the fluid within. The pitfall trap has evolved independently in at least two other groups. The Albany pitcher plant Cephalotus follicularis is a small pitcher plant from Western Australia, with moccasin-like pitchers. The rim of its pitcher's opening is particularly pronounced and provides a thorny overhang to the opening, preventing trapped insects from climbing out. The lining of most pitcher plants is covered in a loose coating of waxy flakes, which are slippery for insects, prey that are often attracted by nectar bribes secreted by the peristome and by bright flower-like anthocyanin patterning. In at least one species, Sarracenia flava, the nectar bribe is laced with cornyrine, a toxic alkaloid also found in hemlock, which probably increases the efficiency of the traps by intoxicating prey. The final carnivore with a pitfall-like trap is the bromeliad Brocchinia reducta. Like most relatives of the pineapple, the tightly packed, waxy leaf bases of the strap-like leaves of this species form an urn. In most bromeliads, water collects readily in this urn and may provide habitats for frogs, insects and, more useful for the plant, disotrophic bacteria. In Brocchinia, the urn is a specialized insect trap, with a loose, waxy lining and a population of digestive bacteria. Flypaper traps. The flypaper trap is based on a sticky mucilage, or glue. The leaf of flypaper traps is studded with mucilage-secreting glands, which may be short, or long and mobile. Flypapers have evolved independently at least 5 times. In the genus Pinguicula, the mucilage glands are quite short, and the leaf, while shiny, does not appear carnivorous. However, this belies the fact that the leaf is an extremely effective trap of small flying insects, and its surface responds to prey by relatively rapid growth. This thymotropic growth may involve rolling of the leaf blade or dishing of the surface under the prey to form a shallow digestive pit. The sundew genus consists of over 100 species of active flypapers whose mucilage glands are borne at the end of long tentacles, which frequently grow fast enough in response to prey to aid the trapping process. The tentacles of D. burmania can bend 180 degrees in a minute or so. Sundews are extremely cosmopolitan and are found on all the continents except the Antarctic mainland. They are most diverse in Australia, the home to the large subgroup of pygmy sundews such as D. pygmaea and to a number of tuberous sundews such as D. peltata, which form tubers that will estivate during the dry summer months. These species are so dependent on insect sources of nitrogen that they generally lack the enzyme nitrate reductase, which most plants require to assimilate soil-borne nitrate into organic forms. Closely related to Drosera is the Portuguese dewy pine, Drosophilum, which differs from the sundews in being passive. Its leaves are incapable of rapid movement or growth. Unrelated, but similar in habit, are the Australian rainbow plants. Drosophilum is unusual in that it grows under near-desert conditions. Almost all other carnivores are either bog plants or grow in moist tropical areas. Recent molecular data indicate that the remaining flypaper, Traphiophyllum peltatum, a member of the Dionchophiliaceae, is closely related to Drosophilum and forms part of a larger clade of carnivorous and non-carnivorous plants with the Droseraceae, Nepenthaceae, Ancestrocladaceae and Plumbaginaceae. This plant is usually encountered as a liana, but in its juvenile phase, the plant is carnivorous. This may be related to a requirement for specific nutrients for flowering. Snap traps. The only two active snap traps are the Venus flytrap and the waterheel plant are believed to have had a common ancestor with similar adaptations. Their trapping mechanism has also been described as a mouse trap, bear trap, or man trap. based on their shape and rapid movement. However, the term snap trap is preferred as other designations are misleading, particularly with respect to the intended prey. Aldrovanda is aquatic and specialized in catching small invertebrates. Dionea is terrestrial and catches a variety of arthropods, including spiders. The traps are very similar, with leaves whose terminal section is divided into two lobes, hinged along the midrib. Trigger hairs inside the trap lobes are sensitive to touch. When a trigger hair is bent, stretch-gated ion channels in the membranes of cells at the base of the trigger hair open, generating an action potential that propagates to cells in the midrib. These cells respond by pumping out ions, which may either cause water to follow by osmosis or cause rapid acid growth. The mechanism is still debated, but in any case, changes in the shape of cells in the midrib allow the lobes, held under tension, to snap shut, flipping rapidly from convex to concave and interring the prey. This whole process takes less than a second. In the Venus flytrap, closure in response to raindrops and blown-in debris is prevented by the leaves having a simple memory. For the lobes to shut, two stimuli are required, 0.5 to 30 seconds apart. The snapping of the leaves is a case of thymonasty. Further stimulation of the lobe's internal surfaces by the struggling insects causes the lobes to close even tighter, sealing the lobes hermetically and forming a stomach in which digestion occurs over a period of 1 to 2 weeks. Leaves can be reused 3 or 4 times before they become unresponsive to stimulation, depending on the growing conditions. Bladder traps. Bladder traps are exclusive to the genus Utricularia, or bladderworts. The bladders pump ions out of their interiors. Water follows by osmosis, generating a partial vacuum inside the bladder. The bladder has a small opening, sealed by a hinged door. In aquatic species, the door has a pair of long trigger hairs. Aquatic invertebrates such as Daphnia touch these hairs and deform the door by lever action, releasing the vacuum. The invertebrate is sucked into the bladder, where it is digested. Many species of Utricularia are terrestrial, growing on waterlogged soil, and their trapping mechanism is triggered in a slightly different manner. Bladderworts lack roots, but terrestrial species have anchoring stems that resemble roots. Temperate aquatic bladderworts generally die back to a resting turion during the winter months, and Eumacroisa appears to regulate the number of bladders it bears in response to the prevailing nutrient content of its habitat. Lobster pot traps. A lobster pot trap is a chamber that is easy to enter, and whose exit is either difficult to find or obstructed by inward-pointing bristles. Lobster pots are the trapping mechanism in Genlycia, the corkscrew plants. These plants appear to specialize in aquatic protozoa. A Y-shaped modified leaf allows prey to enter but not exit. Inward-pointing hairs force the prey to move in a particular direction. Prey entering the spiral entrance that coils around the upper 2 arms of the Y are forced to move inexorably towards a stomach in the lower arm of the Y, where they are digested. Prey movement is also thought to be encouraged by water movement through the trap, produced in a similar way to the vacuum in bladder traps, and probably evolutionarily related to it. Outside of Genlycia, features reminiscent of lobster pot traps can be seen in Sarracenia psittacina, Darlingtonia californica, and, some horticulturalists argue, Nepenthes aristolochioides. Combination traps. The trapping mechanism of the Sundew Drosera glandulata combines features of both flypaper and snap traps. It has been termed a catapult flypaper trap. Borderline carnivores. To be a fully fledged carnivore, a plant must attract, kill, and digest prey. And it must benefit from absorbing the products of the digestion. To many horticulturalists, these distinctions are a matter of taste. There is a spectrum of carnivory found in plants, from completely non-carnivorous plants like cabbages, to borderline carnivores, to unspecialized and simple traps, like Heliumvora, to extremely specialized and complex traps, like that of the Venus flytrap. The borderline carnivores include Virigula and Catopsis berteroniana. Catopsis is a borderline carnivorous bromeliad, like Brocchinia reducta. However, unlike B. reducta, which produces the enzyme phosphatase, C. berteroniana has not been shown to produce any digestive enzymes at all. In these pitfall traps, prey simply fall into the urn, assisted by the waxy scales located on the rim. Arugula has a more intricate relationship with its prey. The plants in this genus produce sticky leaves with resin-tipped glands and look extremely similar to some of the larger sundews. However, they do not directly benefit from the insects they catch. Instead, they form a mutualistic symbiosis with species of assassin bug, which eat the trapped insects. The plant benefits from the nutrients in the bug's feces. A number of species in the Martiniaceae, such as Ebisla lutea, have sticky leaves that trap insects. However, these plants have not been shown conclusively to be carnivorous. Likewise, the seeds of shepherd's purse, urns of Paeopalanthus bromeliodes, bracts of Passiflora fotida, and flower stalks and sepals of trigger plants appear to trap and kill insects, but their classification as carnivores is contentious. The production of specific prey-digesting enzymes is sometimes used as a criterion for carnivory. However, this would probably discount Helianthera and Darlingtonia, both of which appear to rely on the enzymes of symbiotic bacteria to break down their prey but are generally considered as carnivores. However, discounting the enzyme-based definition leaves open the question of Arugula. Evolution. The evolution of carnivorous plants is obscured by the paucity of their fossil record. Very few fossils have been found, and then usually only as seed or pollen. Carnivorous plants are generally herbs, and their traps are produced by primary growth. They generally do not form readily fossilizable structures such as thick bark or wood. The traps themselves would probably not be preserved in any case. Still, much can be deduced from the structure of current traps. Pitfall traps are quite clearly derived from rolled leaves. The vascular tissues of Sarracenia is a case in point. The keel along the front of the trap contains a mixture of leftward and rightward-facing vascular bundles, as would be predicted from the fusion of the edges of an adaxial leaf surface. Flypapers also show a simple evolutionary gradient from sticky, non-carnivorous leaves, through passive flypapers to active forms. Molecular data show the Dianella order of Vanderclade is closely related to Drosera, but the traps are so dissimilar that the theory of their origin very fast-moving flypapers became less reliant on glue remains rather speculative. There are over 250,000 species of flowering plants. Of these, only around 630 are known to be carnivorous. True carnivory has probably evolved independently at least 6 times. However, some of these independent groups probably descended from a recent common ancestor with a predisposition to carnivory. Some groups seem particularly fertile ground for carnivorous pre-adaptation, although in the former case, this may be more to do with the ecology of the group than its morphology, as most of the members of this group grow in low-nutrient habitats such as heath and bog. It has been suggested that all trap types are modifications of a similar basic structure the hairy leaf. Hairy leaves can catch and retain drops of rainwater, especially if shield-shaped or peltate, thus promoting bacteria growth. Insects land on the leaf, become mired by the surface tension of the water, and suffocate. Bacteria jumpstart decay, releasing from the corpse nutrients that the plant can absorb through its leaves. This foliar feeding can be observed in most non-carnivorous plants. Plants that were better at retaining insects or water therefore had a selective advantage. Rainwater can be retained by cupping the leaf, leading to pitfall traps. Alternatively, insects can be retained by making the leaf stickier by the production of mucilage, leading to flypaper traps. The pitfall traps may have evolved simply by selection pressure for the production of more deeply cupped leaves, followed by zipping up of the margins and subsequent loss of most of the hairs, except at the bottom, where they help retain prey. The lobster pot traps of Genlycia are difficult to interpret. They may have developed from bifurcated pitchers that later specialized on ground-dwelling prey. Or, perhaps, the prey-guiding protrusions of bladder traps became more substantial than the net-like funnel found in most aquatic bladderworts. Whatever their origin, the helical shape of the lobster pot is an adaptation that displays as much trapping surface as possible in all directions when buried in moss. The traps of the bladderworts may have derived from pitchers that specialized in aquatic prey when flooded, like Sarracenia psittacina does today. Escaping prey and terrestrial pitchers have to climb or fly out of a trap, and both of these can be prevented by wax, gravity and narrow tubes. However, a flooded trap can be swum out of, so in Utricularia, a one-way lid may have developed to form the door of a protobladder. Later, this may have become active by the evolution of a partial vacuum inside the bladder, tripped by prey brushing against trigger hairs on the door of the bladder. Flypaper traps include the various true flypapers and the snap traps of Aldrovanda and Dionea. The production of sticky mucilage is found in many non-carnivorous genera, and the passive glue traps in Bibeli and Drosophilum could easily have evolved. The active glue traps use rapid plant movements to trap their prey. Rapid plant movement can result from actual growth, or from rapid changes in cell turgor, which allow cells to expand or contract by quickly altering their water content. Slow-moving flypapers like Pinguicula exploit growth, but the Venus flytrap uses such rapid turgor changes that glue became unnecessary. The stalked glands that once made it and which are so evident in Drosera have become the teeth and trigger hair, so an example of natural selection hijacking pre-existing structures for new functions. Recent taxonomic analysis of the relationships within the Chiophilales indicate that the Droseraceae, Trerpiopilum, Nepentacea and Drosophilum, while closely related, are embedded within a larger clade that includes non-carnivorous groups such as the tamarisks, Ancestrocladaceae, Polygomnaceae and Plumbaginaceae. Interestingly, the tamarisks possess specialized salt-excreting glands on their leaves, as do several of the Plumbaginaceae, which may have been co-opted for the excretion of other chemicals, such as proteases and mucilage. Some of the Plumbaginaceae also have stalked, vascularized glands that secrete mucilage on their calyces and aid in seed dispersal and possibly in protecting the flowers from crawling parasitic insects. These are probably homologous with the tentacles of the carnivorous genera. Perhaps carnivory evolved from a protective function, rather than a nutritional one. The balsams, which are closely related to the Sarraceniaceae and Rorigula, similarly possess stalked glands. The only traps that are unlikely to have descended from a hairy leaf or sepal are the carnivorous bromeliads. These plants use the urna, a fundamental part of a bromeliada, for a new purpose and build on it by the production of wax and the other paraphernalia of carnivory. Ecology and modeling of carnivory. Carnivorous plants are widespread but rather rare. They are almost entirely restricted to habitats such as bogs, where soil nutrients are extremely limiting, but where sunlight and water are readily available. Only under such extreme conditions is carnivory favored to an extent that makes the adaptations obvious. The archetypal carnivore, the Venus flytrap, grows in soils with almost immeasurable nitrate and calcium levels. Plants need nitrogen for protein synthesis, calcium for cell wall stiffening, phosphate for nucleic acid synthesis, and iron for chlorophyll synthesis. The soil is often waterlogged, which favors the production of toxic ions such as ammonium, and its pH is an acidic 4 to 5. Ammonium can be used as a source of nitrogen by plants, but its high toxicity means that concentrations high enough to fertilize are also high enough to cause damage. However, the habitat is warm, sunny, constantly moist, and the plant experiences relatively little competition from low-growing sphagnum moss. Still, carnivores are also found in very atypical habitats. Drosophilum lusitanicum is found around desert edges and Pinguicula vallisneriifolia on limestone cliffs. In all the studied cases, carnivory allows plants to grow and reproduce using animals as a source of nitrogen, phosphorus and possibly potassium. However, there is a spectrum of dependency on animal prey. Pygmy sundews are unable to use nitrate from soil because they lack the necessary enzymes. Common butterworts can use inorganic sources of nitrogen better than organic sources, but a mixture of both is preferred. European bladderworts seem to use both sources equally well. Animal prey makes up for differing deficiencies in soil nutrients. Plants use their leaves to intercept sunlight. The energy is used to reduce carbon dioxide from the air with electrons from water to make sugars and a waste product, oxygen, in the process of photosynthesis. Leaves also respire, in a similar way to animals, by burning their biomass to generate chemical energy. This energy is temporarily stored in the form of ATP, which acts as an energy currency for metabolism in all living things. As a waste product, respiration produces carbon dioxide. For a plant to grow, it must photosynthesize more than it respires. Otherwise, it will eventually exhaust its biomass and die. The potential for plant growth is net photosynthesis, the total gross gain of biomass by photosynthesis, minus the biomass lost by respiration. Understanding carnivory requires a cost-benefit analysis of these factors. In carnivorous plants, the leaf is not just used to photosynthesize, but also as a trap. Changing the leaf shape to make it a better trap generally makes it less efficient at photosynthesis. For example, pitchers have to be held upright, so that only their opercula directly intercept light. The plant also has to expend extra energy on non-photosynthetic structures like glands, hairs, glue and digestive enzymes. To produce such structures, the plant requires ATP and respires more of its biomass. Hence, a carnivorous plant will have both decreased photosynthesis and increased respiration, making the potential for growth small and the cost of carnivory high. Being carnivorous allows the plant to grow better when the soil contains little nitrate or phosphate. In particular, an increased supply of nitrogen and phosphorus makes photosynthesis more efficient, because photosynthesis depends on the plant being able to synthesize very large amounts of the nitrogen-rich enzyme rubisco, the most abundant protein on Earth. It is intuitively clear that the Venus flytrap is more carnivorous than Traphiophyllum peltatum. The former is a full-time moving snap trap. The latter is a part-time, non-moving flypaper. The energy wasted by the plant in building and fueling its trap is a suitable measure of the carnivory of the trap. Using this measure of investment in carnivory, a model can be proposed. Above is a graph of carbon dioxide uptake against trap respiration for a leaf in a sunny habitat containing no soil nutrients at all. Respiration is a straight line sloping down under the horizontal axis. Gross photosynthesis is a curved line above the horizontal axis. As investment increases, so too does the photosynthesis of the trap, as the leaf receives a better supply of nitrogen and phosphorus. Eventually another factor will become more limiting to photosynthesis than nitrogen or phosphorus supply. As a result, increasing the investment will not make the plant grow better. The net uptake of carbon dioxide, and therefore, the plant's potential for growth, must be positive for the plant to survive. There is a broad span of investment where this is the case, and there is also a non-zero optimum. Plants investing more or less than this optimum will take up less carbon dioxide than an optimal plant, and hence growing less well. These plants will be at a selective disadvantage. At zero investment the growth is zero, because a non-carnivorous plant cannot survive in a habitat with absolutely no soil-borne nutrients. Such habitats do not exist, so for example, sphagnum absorbs the tiny amounts of nitrates and phosphates in rain very efficiently and also forms symbioses with disotrophic cyanobacteria. In a habitat with abundant soil nutrients but little light, the gross photosynthesis curve will be lower and flatter, because light will be more limiting than nutrients. A plant can grow at zero investment in carnivory. This is also the optimum investment for a plant, as any investment in traps reduces net photosynthesis to less than the net photosynthesis of a plant that obtains its nutrients from soil alone. Carnivorous plants exist between these two extremes. The less limiting light and water are, and the more limiting soil nutrients are, the higher the optimum investment in carnivory, and hence the more obvious the adaptations will be to the casual observer. The most obvious evidence for this model is that carnivorous plants tend to grow in habitats where water and light are abundant and where competition is relatively low, the typical bog. Those that do not tend to be even more fastidious in some other way. Drosophilum lusitanicum grows where there is little water, but it is even more extreme in its requirement for bright light and low disturbance than most other carnivores. Pinguicula Evolutionary grows in soils with high levels of calcium but requires strong illumination and lower competition than many butterworts. In general, carnivorous plants are poor competitors, because they invest too heavily in structures that have no selective advantage in nutrient-rich habitats. They succeed only where other plants fail. Carnivores are to nutrients what cacti are to water. Carnivory only pays off when the nutrient stress is high and where light is abundant. When these conditions are not met, some plants give up carnivory temporarily. Sarracenia spp produce flat, non-carnivorous leaves in winter. Light levels are lower than in summer, so light is more limiting than nutrients, and carnivory does not pay. The lack of insects in winter exacerbates the problem. Damage to growing pitcher leaves prevent them from forming proper pitchers, and again, the plant produces a phyllodes instead. Many other carnivores shut down in some seasons. Tuberous sundews die back to tubers in the dry season, bladderworts to turians in winter, and non-carnivorous leaves are made by most butterworts and cephalotus in the less favorable seasons. Utricularia macroeza varies the number of bladders it produces based on the expected density of prey. Part-time carnivory in Tropiophyllum peltatum may be due to an unusually high need for potassium at a certain point in the life cycle, just before flowering. The more carnivorous a plant is, the less conventional its habitat is likely to be. Venus flytraps live in a very specialized habitat, whereas less carnivorous plants are found in less unusual habitats. Bibeli and Drosophilum both come from relatively arid regions and are both passive flypapers, arguably the lowest maintenance form of trap. Venus flytraps filter their prey using the teeth around the trap's edge, so as not to waste energy on hard-to-digest prey. In evolution, laziness pays, because energy can be used for reproduction, and short-term benefits in reproduction will outweigh long-term benefits in anything else. Carnivory rarely pays, so even carnivorous plants avoid it when there is too little light or an easier source of nutrients, and they use as few carnivorous features as are required at a given time or for a given prey item. There are very few habitats stressful enough to make investing biomass and energy in trigger hairs and enzymes worthwhile. Many plants occasionally benefit from animal protein rotting on their leaves, but carnivory that is obvious enough for the casual observer to notice is rare. Bromeliads seem very well pre-adapted to carnivory, but only one or two species can be classified as truly carnivorous. By their very shape, bromeliads will benefit from increased prey-derived nutrient input. In this sense, bromeliads are probably carnivorous, but their habitats are too dark for more extreme, recognizable carnivory to evolve. Most bromeliads are epiphytes, and most epiphytes grow in partial shade on tree branches. Brooksgenia reducta, on the other hand, is a ground dweller. Many carnivorous plants are not strongly competitive and rely on circumstances to suppress dominating vegetation. Accordingly, some of them rely on fire ecology for their continued survival. For the most part, carnivorous plant populations are not dominant enough to be dramatically significant, ecologically speaking, but there is an impressive variety of organisms that interact with various carnivorous plants in sundry relationships of kleptoparasitism, commensalism, and mutualism. For example, small insectivores such as tree frogs often exploit the supply of prey to be found in pitcher plants, and the frog Microelenipenthicola actually specializes in such habitats. Certain crab spiders such as Musumenips nepenthicola live largely on the prey of nepenthes, and other, less specialized, spiders may build webs where they trap insects attracted by the smell or appearance of the traps. Some scavengers, detritivores, and also organisms that harvest or exploit those in turn, such as the mosquito Wyomysmithia, are largely or totally dependent on particular carnivorous plants. Plants such as Rigula species combine with specialized bugs in benefiting from insects trapped on their leaves. Associations with species of pitcher plants are so many and varied that the study of nepenthes in fauna is something of a discipline in its own right. Campanopus schmidtsi, the diving ant, has an intimate degree of mutualism with the pitcher plant Nepenthes bicalcarata. It not only retrieves prey and detritus from beneath the surface of the liquid in the pitchers, but repels herbivores, and cleans the pitcher peristome, maintaining its slippery nature. The ants have been reported to attack struggling prey, hindering their escape, so there might be an element of mimicotrophy to the relationship. Numerous species of mosquitoes lay their eggs in the liquid, where their larvae play various roles, depending on species. Some eat microbes and detritus, as is common among mosquito larvae, whereas some species of toxorynchids also breed in pitchers, and their larvae are predators of other species of mosquito larvae. Apart from the crab spiders on pitchers, an actual small, red crab Geosesamum malayanum will enter the fluid, robbing and scavenging, though reputedly it does so at some risk of being captured and digested itself. Nepenthes raja has a remarkable mutualism with two unrelated small mammals, the tree shrew Tupaia montana and the summit rat, Raptors boluensis. The tree shrews and the rats defecate into the plant's traps while visiting them to feed on sweet, fruity secretions from glands on the pitcher lids. The tree shrew also has a similar relationship with at least two other giant species of Nepenthes. More subtly, Hardwick's woolly bat, a small species, roosts beneath the operculum of Nepenthes hemscheliana. The bat's excretions that land in the pitcher pay for the shelter, as it were. To the plant the excreta are more readily assimilable than intact insects would be. There also is a considerable list of Nepenthes endophytes. These are microbes other than pathogens that live in the tissues of pitcher plants, often apparently harmlessly. Classification. The classification of all flowering plants is currently in a state of flux. In the Cronquist system, the Droseraceae and Nepenthaceae were placed in the order Nepenthes, based on the radial symmetry of their flowers and their possession of insect traps. The Sarraceniaceae was placed either in the Nepenthes, or in its own order, the Sarraceniales. The Biblodaceae, Cephalataceae, and Rorigulaceae were placed in the Saxifragales, and the Lentibulariaceae in the Scrofulariales. In more modern classification, such as that of the Angiosperm phylogeny group, the families have been retained, but they have been redistributed amongst several disparate orders. It is also recommended that Drosophilum be considered in a monotypic family outside the rest of the Droseraceae, probably more closely allied to the Dionchophyllaceae. The current recommendations are shown below. Dicots. Asterales, Stylidiaceae, Stylidium. Chaeophyllales, Dionchophyllaceae, Trerphiophyllum. Drosophiliaceae, Drosophyllum. Droseraceae, Aldrovanda, Dionea, Drosera, Eurodroseropolis, Eurodroseropids, Eurodroserodites, Eurofisharopolis, Europaleoaldrovanda, Eurosaxonopolis. Nepenthaceae, Nepenthes. Ericales, Perigulaceae, Perigula. Sarraceniaceae, Euroarcanivora, Sarracenia, Darlingtonia, Helianthera. Lamiales, Biblodaceae, Bibili. Lentibulariaceae, Pincuicula, Genlycea, Utricularia. Martiniaceae, Ebisla. Oxalidales, Cephalotus. Monocots, Poles, Bromeliaceae, Brachynia, Catopsis. Eriosolicaceae, Paepalanthus bromeliodes. Cultivation. Although different species of carnivorous plants have different requirements in terms of sunlight, humidity, soil moisture, etc., there are commonalities. Most carnivorous plants require rainwater, or water that has been distilled, deionized by reverse osmosis, or acidified to around penthouse 600 using sulfuric acid. Common tap or drinking water contains minerals that will quickly build up and kill the plant. This is because most carnivorous plants have evolved in nutrient-poor, acidic soils and are consequently extreme calcifuges. They are therefore very sensitive to excessive soil-borne nutrients. Since most of these plants are found in bogs, almost all are very intolerant of drying. There are exceptions. Tuberous sundews require a dry dormancy period, and Drosophilum requires much drier conditions than most. Outdoor-grown carnivorous plants generally catch more than enough insects to keep themselves properly fed. Insects may be fed to the plants by hand to supplement their diet. However, carnivorous plants are generally unable to digest large non-insect food items. Bits of hamburger, for example, will simply rot, and this may cause the trap, or even the whole plant, to die. A carnivorous plant that catches no insects at all will rarely die, although its growth may be impaired. In general, these plants are best left to their own devices. After underwatering with tap water, the most common cause of Venus flytrap death is prodding the traps to watch them close and feeding them inappropriate items. Most carnivorous plants require bright light, and most will look better under such conditions, as this encourages them to synthesize red and purple anthocyanin pigments. Nepenthes and Pinguicula will do better out of full sun, but most other species are happy in direct sunlight. Carnivores mostly live in bogs, and those that do not are generally tropical. Hence, most require high humidity. On a small scale, this can be achieved by placing the plant in a wide saucer containing pebbles that are kept permanently wet. Small Nepenthes species grow well in large terraria. Many carnivores are native to cold temperate regions and can be grown outside in a bog garden year-round. Most Sarracenia can tolerate temperatures well below freezing, despite most species being native to the southeastern United States. Species of Drosera and Pinguicula also tolerate sub-freezing temperatures. Nepenthes species, which are tropical, require temperatures from 20 to 30 degrees Celsius to thrive. Carnivorous plants require appropriate nutrient-poor soil. Most appreciate a 3-1 mixture of sphagnum peat to sharp horticultural sand. Nepenthes will grow in orchid compost or in pure sphagnum moss. Ironically, carnivorous plants are themselves susceptible to infestation by parasites such as aphids or mealybugs. Although small infestations can be removed by hand, larger infestations necessitate use of an insecticide. Isopropyl alcohol is effective as a topical insecticide, particularly on scale insects. Diazinon is an excellent systemic insecticide that is tolerated by most carnivorous plants. Malathion and esophate have also been reported as tolerable by carnivorous plants. Although insects can be a problem, by far the biggest killer of carnivorous plants is grey mold. This thrives under warm, humid conditions and can be a real problem in winter. To some extent, temperate carnivorous plants can be protected from this pathogen by ensuring that they are kept cool and well ventilated in winter and that any dead leaves are removed promptly. If this fails, a fungicide is in order. The easiest carnivorous plants for beginners are those from the cool temperate zone. These plants will do well under cool greenhouse conditions if kept in wide trays of acidified or rainwater during summer and kept moist during winter. Drosera capensis, the Cape sundew, attractive strap-leaved sundew, pink flowers, very tolerant of maltreatment. Drosera binata, the fork-leaved sundew, large, Y-shaped leaves. Sarracenia flava, the yellow trumpet pitcher, yellow, attractively veined leaves, yellow flowers in spring. Pinguicula grandiflora, the common butterwort, purple flowers in spring, hibernates as a bud in winter. Fully hardy. Pinguicula moronensis, the Mexican butterwort, pink flowers, non-carnivorous leaves in winter. Venus flytraps will do well under these conditions but are actually rather difficult to grow. Even if treated well, they will often succumb to grey mold in winter unless well ventilated. Some of the lowland Nepenthes are very easy to grow as long as they are provided with relatively constant, hot and humid conditions. Medicinal uses. A study published in 2009 by researchers from Tel Aviv University indicates that secretions produced by carnivorous plants contain compounds that have antifungal properties and may lead to the development of a new class of antifungal drugs that will be effective against infections that are resistant to current antifungal drugs. Cultural depictions. Carnivorous plants have long been the subject of popular interest and exposition, much of it highly inaccurate. Fictional plants have been featured in a number of books, movies, television series, and video games. Typically, these fictional depictions include exaggerated characteristics, such as enormous size or possession of abilities beyond the realm of reality, and can be viewed as a kind of artistic license. Two of the most famous examples of fictional carnivorous plants in popular culture are the 1960s black comedy The Little Shop of Horrors and the Triffids of John Wyndham's The Day of the Triffids. Other movies, such as The Hellstrom Chronicle, and television series utilize accurate depictions of carnivorous plants for cinematic purposes. The earliest known depiction of carnivorous plants in popular culture was a case wherein a large man-eating tree was reported to have consumed a young woman in Madagascar in 1878, as witnessed by Dr. Carl Leach. Leach reported the events in the South Australian Register in 1881. The woman, pictured in an accompanying artwork, was supposed to have been a member of the Mkodos, a little-known but cruel tribe. The account has been debunked as pure myth as it appears Dr. Leach, the Mkodos, and the tree were all fabrications. See also: aggressive mimicry, carnivorous fungus, predatory dinoflagellate, references. Further reading: Slack A. Insect-eating plants and how to grow them. Sherborne, UK, Alphabooks. ISBN A0-906670-42-Exeter, Juniper B.E., Robbins R.J., Joel Dam. The Carnivorous Plants. Academic Press, San Diego. A carnivorous plant database provides an up-to-date, searchable database of all the published species of carnivorous plants. Carnivorous Plant FAQ at saracenia.com, list of films and TV shows that feature carnivorous plants, most of them fictional. Botanical Society of America, Carnivorous Plants Online, Inner World of Carnivorous Plants from the John Innes Center, Ellison, AM 2006. Nutrient Limitation and Stoichiometry of Carnivorous Plants, a PDF, 334 KB, Plant Bio 8, 740 €747.